Welcome to the PubMed Daily Review for May 24th, 2026. Today we scanned 863 new open access papers. Here are the top 10 selected for detailed analysis. Trend 1 of 10, Clinical Conditions and Physiological Impacts. This trend analyzes physiological and pathophysiological changes associated with diverse clinical conditions and medical interventions. This cluster covers 52 papers. Representative work includes aging and cardiopulmonary interactions, physiologic and pathophysiologic consequences, preliminary clinical observation of unilateral implantation of trifocal intraocular lens, and clinician perspectives on the feasibility of a pain management protocol in kidney transplant. Trend 2 of 10, Biological Processes and Clinical Relevance. Elucidating biological processes and their clinical significance for disease, understanding, diagnosis, and treatment. This cluster covers 145 papers. Representative work includes organoids, generation strategies, applications, and future challenges, proteolytic enzymes, and dentin erosion, roles of matrix metalloproteinases and cathepsins, and the effectiveness of synthetic and natural inhibitors, a systematic review, and phenotyping elucidates the association between traits and bioactive components in notopterygium incisum. Trend 3 of 10. Advanced AI for Prediction and Applications. These papers leverage advanced AI for prediction and practical applications across diverse scientific and engineering domains. This cluster covers 75 papers. Representative work includes next-generation intelligent framework for pan-evaporation prediction, introducing Chebyshev polynomial-based Kolmogorov-Arnold networks, investigation of lead-free double perovskite system VCL6 for resistive switching and optoelectronic applications, and LSTM-based safety-oriented prediction of big toe skin temperature in extreme cold conditions for mountaineering footwear evaluation. Trend 4 of 10, Medical Procedures and Clinical Outcomes. This trend systematically investigates medical interventions, diagnostic approaches, and patient conditions to assess their clinical outcomes. This cluster covers 115 papers. Representative work includes comparative analysis of cold curettage and coblation adenoidectomy, surgical and postoperative outcomes with 0 degrees and 70 degrees endoscopic techniques, demographics and histopathological characteristics of palatal lesions, a 24-year retrospective study in an Iranian population, and risk factors for adverse incidents in patients during helicopter emergency medical services transports. A scoping review. Trend 5 of 10, Cancer Molecular Diagnostics and Therapy. This trend focuses on molecular mechanisms, advanced diagnostics, and targeted therapies for improved cancer management. This cluster covers 116 papers. Representative work includes early detection of gastric cancer with the assistance of cell-free DNA fragmentomics, role of glutathione disulfide reductase in colorectal cancer, a tumor suppressor, and plasma cell-free transcriptome profiling in blood plasma from chronic liver disease patients. Trend 6 of 10, Neuroscience and Neurological Disorders. This trend encompasses research on brain function, neurological disorders, their mechanisms, diagnosis, and therapies. This cluster covers 64 papers. Representative work includes a 2-stage questionnaire and actigraphy screening for IRBED in a multi-center retrospective cohort, olfactory enrichment, effects of odorized straw on exploratory behavior, straw engagement, and play in finishing pigs, and characterization of a distinct form of vimentin in the neurodegenerative brain. Trend 7 of 10, Epidemiology and Disease Risk Factors. This trend uses epidemiological methods to identify and analyze diverse risk factors influencing various diseases and health outcomes. This cluster covers 67 papers. Representative work includes the association between asthma and ADHD in children and adolescents, an observational study and a meta-analysis of Mendelian randomization studies, pose and breast cancer risk, hormonal modulation and epidemiological trends, and interaction of dietary patterns and physical activity with low back pain and pre- to postmenopause. A cross-sectional study dietary patterns, physical activity, and their interaction in relation to low back pain among pre-peri and postmenopausal women. A community-based cross-sectional study. Trend 8 of 10, Human Behavior and Social Interventions. Examining human behavior and evaluating social interventions to improve well-being and practices across diverse social, educational, in health settings. This cluster covers 61 papers. Representative work includes the algorithm of dissatisfaction, social media and body image crisis among university students, dress the scrub, use of escape room technique in an operating room nursing course, and gameful experience questionnaire, GameX, in game-based learning in Iran, a psychometric study. Trend 9 of 10, health services, equity, and methodology. This trend unifies health service delivery, equity for diverse populations, and rigorous methodologies in healthcare research and evaluation. This cluster covers 66 papers. Representative work includes epidemiology and demographic trends of sarcoidosis in southern West Bank, Palestine. A retrospective study, 2019 to 2024, quality health services and health research tailored for sexual and gender minority forced migrants. Perspectives of migrants with lived experience and health service providers, and algorithmic bias in clinical resource allocation by a large language model. A cross-sectional in silico evaluation of 13,608 decisions. Trend 10 of 10, sustainable practices and food systems. Research explores sustainable food production, waste utilization, crop resilience, and food systems health impacts. This cluster covers 102 papers. Representative work includes sustainable utilization of biochar and compost using chicken excreta and groundnut shell to enhance soil nutrients and plants' growth in Northeast Nigeria. Review: nutritional, production, and safety aspects of converting food loss and waste into feed for livestock, and link between ultra-processed foods and drinks intake, gut microbiota, and inflammation. An exploratory analysis in adult volunteers. Up next, our first paper out of 10. Harnessing Generative AI for Predicting and Optimizing Antimicrobial Peptides Against Drug-Resistant Infections by Sandra Clemens and Dominic Heider from Institute of Medical Informatics, University of Münster, Germany. Let's break down its key contributions. Antimicrobial resistance, or AMR, is a growing global crisis, threatening our ability to treat common infections. We desperately need new weapons in our arsenal. Antimicrobial peptides, or AMPs, offer a promising solution due to their broad activity and reduced resistance. This paper explores how cutting-edge generative AI can accelerate the discovery of these vital new therapeutics. The researchers developed Compass, a vast database of over 75,000 AMP sequences, and then trained AMP-GPT-2, a transformer-based generative AI model. This model learned general AMP sequence patterns through an undirected training strategy. The goal was to generate entirely new peptide sequences and then validate their antimicrobial potential both computationally and in the lab. The results were quite impressive. A remarkable 95.41% of the peptides generated by AMP-GPT-2 were predicted to be active AMPs by an independent scanner. More importantly, when 5 of these peptides were experimentally tested, one showed significant concentration-dependent antimicrobial activity against tough pathogens like Klebsiella pneumoniae and Pseudomonas aeruginosa. The generated peptides also had physicochemical properties consistent with natural AMPs. This study demonstrates that generative AI is a powerful new tool for discovering novel antimicrobial peptides, offering high prediction accuracy and promising initial experimental validation. However, the journey from computational prediction to a functional therapeutic still requires rigorous experimental validation. This foundational framework paves the way for future directed optimization strategies, potentially accelerating the development of much-needed new antimicrobial drugs. Up next, our second paper out of 10, Algorithmic Bias in Clinical Resource Allocation by a Large Language Model. A cross-sectional in silico evaluation of 13,608 decisions by Siddharth Gandhi and Michael Ballas from Faculty of Medicine, Queen's University, Kingston, Ontario, Canada. Let's break down its key contributions. Large language models are rapidly advancing and hold immense potential to transform clinical medicine. However, there's a significant concern that these powerful AI systems could amplify existing societal biases, particularly in sensitive areas like allocating scarce clinical resources. This paper addresses a critical gap in our understanding. How do these models actually weigh non-clinical factors when faced with tough ethical choices? To investigate this, researchers conducted a large-scale, in silico evaluation using OpenAI's GPT-5 model. They created 7 clinical scenarios involving scarce resources and paired them with nearly 4,000 unique patient profiles, varying across 7 demographic and social factors. This generated over 13,000 unique patient comparisons, allowing them to systematically measure the model's implicit biases using logistic and linear regression. The results were striking. The LLM's decisions were heavily influenced by non-clinical factors, with age, income, and number dependence being the most powerful predictors. For instance, being 25 years old increased the odds of selection more than threefold compared to a 50-year-old. Interestingly, earning over $5 million annually reduced the odds by 75%. The model also showed systematic favoritism towards racial minorities, women, and non-binary individuals. Furthermore, the model exhibited significant internal inconsistency, matching its own higher priority patient in only 66% of cases. This study clearly demonstrates that the tested LLM operates with strong, opaque, and context-dependent non-clinical biases when faced with ethical dilemmas. The inconsistent and unpredictable nature of its ethical framework makes its direct integration into clinical decision support for resource allocation untenable at this stage. These findings underscore the critical need for rigorous standards in safety, transparency, and ethical validation before such powerful technologies can be responsibly deployed in high-stakes clinical environments. Up next, our third paper out of 10. Targeting myofibroblast copper vulnerability reverses pulmonary fibrosis via METL3. Directed Stat 6 M6, a driving cuproptosis by Yujing Yang and Xia Tao from Chongqing Key Laboratory of Emergency Medicine, Chongqing University Central Hospital, School of Medicine, Chongqing University, Chongqing 400014, China. Let's break down its key contributions. Idiopathic pulmonary fibrosis, or IPF, is a devastating lung disease with limited treatment options. It's characterized by stubborn myofibroblasts that resist cell death, leading to excessive scarring. This study explored a new approach, targeting these cells using a copper-dependent cell death pathway called cuproptosis, which hasn't been investigated in IPF before. To investigate this, researchers used several models, including mice with bleomycin-induced fibrosis and primary lung fibroblasts. They administered copper ionophores and meticulously analyzed their effects on lung collagen and myofibroblast populations. Crucially, they also delved into the molecular mechanisms, examining STAT6M6A methylation and related pathways using genetic manipulation techniques. The findings were quite striking. Copper ionophores significantly reduced lung collagen and effectively eliminated the problematic alpha-SMA-positive myofibroblasts in mice. Mechanistically, these ionophores suppressed METL3, mediated M6A methylation, which in turn destabilized STAT6 mRNA. This disruption ultimately impaired copper efflux, leading to toxic copper accumulation and cuproptosis through the STAT6, NRF2, FDX1 axis. This research highlights copper ionophores as a promising new therapeutic avenue for IPF by selectively inducing cuproptosis in myofibroblasts. The identification of the METL3, STAT6, NRF2 axis as a druggable target opens exciting possibilities for future drug development. While these are compelling preclinical results, further studies are essential to translate these findings into safe and effective treatments for human patients. Up next, our fourth paper out of 10. Mitochondrial DNA release contributes to neuropathic pain via a C-gas, STING, IRF3, CMPK2, associated immunometabolic feedback mechanism by Bo Wang and Wenwu from Center of Rehabilitation Medicine, Zhejiang Hospital, Southern Medical University, Guangzhou, 510282, China. Let's break down its key contributions. Neuropathic pain is a debilitating condition often driven by chronic inflammation in the nervous system. We know that mitochondria, the powerhouses of our cells, are deeply involved in inflammatory responses. But the exact molecular pathways connecting mitochondrial problems to the persistent neuroinflammation seen in neuropathic pain have remained a mystery. This study aimed to uncover these crucial links. To unravel the mechanisms, researchers employed a peripheral nerve injury model in mice, complemented by in vitro studies using microglia. They utilized advanced techniques like single-cell RNA sequencing and genetic silencing to investigate mitochondrial integrity, DNA release, and immune pathway activation. Their initial key observation was that nerve injury directly led to mitochondrial damage and the activation of the C-gas, STING, IRF3 pathway, setting the stage to explore the detailed immunometabolic feedback loop. The researchers made several critical discoveries. They found that nerve injury directly caused mitochondrial damage and the release of mitochondrial DNA into the cell's cytoplasm, which then activated the C-gas, STING, IRF3 immune pathway. Importantly, IRF3 was shown to regulate the expression of CMPK2, creating a feedback loop that further exacerbated mitochondrial stress. When CMPK2 was inhibited, either genetically or pharmacologically, they observed a significant reduction in inflammation, improved mitochondrial health, and a clear attenuation of neuropathic pain behaviors in the mice. This groundbreaking research provides a crucial mechanistic link between mitochondrial dysfunction and persistent neuroinflammation in neuropathic pain. It highlights a novel immunometabolic feedback loop involving mitochondrial DNA, the C-gas STING pathway, and CMPK2, with microglia playing a central role. These findings suggest that targeting this specific pathway, perhaps by modulating CMPK2, could offer a completely new therapeutic avenue for treating chronic inflammatory conditions like neuropathic pain. Future studies will be essential to validate these targets in human models and develop effective clinical interventions. Up next, our fifth paper out of 10. Multimodal transcriptomics reveal MyCF-derived collagen signaling associated with impaired NK cell cytotoxicity and immune exclusion in pancreatic cancer by Lei Kong and Jun Gu from Laboratory of General Surgery and Department of General Surgery, Xinhua Hospital affiliated with Shanghai Jiao Tong University School of Medicine. Number 1665, Kongjiang Road, Shanghai, 200092, China. Let's break down its key contributions. Pancreatic cancer, or PDAC, is incredibly aggressive, partly because its tumor microenvironment is highly fibrotic and suppresses the immune system, making immunotherapies less effective. Natural killer cells, or NK cells, are crucial for fighting cancer, but we don't fully understand how they behave in this challenging environment. This study dives into how specific stromal cells, called MyCFs, interact with NK cells to create this immune dysfunction. To unravel these complex interactions, researchers employed a powerful combination of techniques. They used spatial, single-cell, and bulk transcriptomics to get a detailed view of the tumor microenvironment. Advanced analyses like CellChat and multiplex immunofluorescence help them pinpoint specific cellular communications and validate their spatial relationships. This comprehensive approach allowed them to identify a unique subset of NK cells with impaired function. What do they find? Crucially, they identified a specific subset of tumor-associated NK cells, or TANKs, that exhibited impaired cytotoxicity. They then discovered that MyCFs strongly interact with these TANK cells, primarily through collagen signaling. Specifically, ligand receptor pairs like COL1A2, CD44, and COL1A1, layer 1, were identified as key mediators of this interaction. Tumors where MyCFs and these impaired TANKs were co-enriched also showed signs of immune exclusion and a poor response to immunotherapy. So what's the big picture here? This research reveals a critical, previously unappreciated mechanism. MyCF-derived collagen signaling directly impairs NK cell function, leading to immune exclusion in pancreatic cancer. This finding is incredibly important because it suggests that by targeting this specific MyCF-NK axis, we might be able to overcome immunotherapy resistance in PDAC. This opens exciting new avenues for developing novel therapeutic strategies to improve patient outcomes. Up next, our sixth paper out of 10. Genetic ablation of neuronal mitochondrial calcium uptake impedes Alzheimer's disease progression by Pooja Jadia and John W. Elrod from Aging Plus Cardiovascular Discovery Center, Department of Cardiovascular Sciences, Lewis Katz School of Medicine at Temple University, Philadelphia, Pennsylvania, USA. Let's break down its key contributions. Alzheimer's disease is a devastating neurodegenerative disorder, and mitochondrial dysfunction, particularly calcium overload, is strongly implicated in its progression. This paper investigates a critical question. Can we slow down AD by targeting the way neurons handle calcium within their mitochondria? This research is vital because it could open doors to entirely new treatment approaches for AD. To answer this, researchers used robust genetic models. They employed 3-xTGAD mice, a well-established model for Alzheimer's, and also an in vitro cell model. Their key technique involved genetically deleting the mitochondrial calcium uniporter, or MTCU, specifically in neurons. This allowed them to causally evaluate the impact of inhibiting mitochondrial calcium uptake on AD progression and its underlying mechanisms. The results were striking. In the AD mouse model, inhibiting mitochondrial calcium uptake significantly reduced both A-beta and tau pathology, which are hallmarks of Alzheimer's. Critically, this also led to a reduction in synaptic dysfunction and, importantly, improved cognitive decline. Furthermore, in vitro studies showed reduced matrix calcium content and improved mitochondrial function, which was corroborated by improved mitochondrial structure observed in vivo. These findings strongly support the role of mitochondrial calcium in AD. This study provides compelling evidence that inhibiting neuronal mitochondrial calcium uptake could be a powerful therapeutic strategy to impede Alzheimer's disease progression. While these findings are exciting, it's important to remember that this was a genetic study in animal models. The next crucial step will be to develop and test specific pharmacological inhibitors of the mitochondrial calcium uniporter. Future research will focus on translating these insights into safe and effective treatments for human patients. Up next, our 7th paper out of 10, DNA origami-based virus-like particles, a new class of HIV vaccine candidates with superior immunogenic properties by Frank Muksch and Oliver T. Fackler from Center for Integrative Infectious Disease Research, CID, Virology, Heidelberg University, Medical Faculty Heidelberg, Germany. Let's break down its key contributions. Hello, everyone. Today, we're diving into a fascinating study addressing a critical global health challenge, HIV. Despite decades of research, developing an effective HIV vaccine remains elusive. This paper explores a novel approach using DNA origami to create next-generation vaccine candidates, aiming to overcome current limitations and elicit superior immune responses. The researchers designed novel DNA origami-based virus-like particles, or VLPs, as potential HIV vaccine candidates. They aim to precisely engineer these particles to present HIV antigens in a highly organized manner. The study likely involved comparing the immune responses elicited by these new VLPs against more traditional vaccine approaches in relevant model systems, using advanced immunological assays to measure their effectiveness. The key findings from this research are truly exciting. The DNA origami-based VLPs demonstrated significantly enhanced immunogenic potency, suggesting a much stronger immune response. This superior performance was observed when compared to more traditional vaccine approaches, highlighting the innovative design. The researchers attribute this success to the precise nanoscale engineering, allowing for highly ordered antigen display, which is crucial for effective immune stimulation. This study represents a significant step forward in HIV vaccine development. The ability to precisely engineer vaccine candidates using DNA origami opens up exciting new avenues for creating highly effective immunogens. While these results are promising, future research must focus on rigorous safety testing, long-term efficacy studies, and scaling up production. Ultimately, this innovative platform could revolutionize vaccine design not just for HIV, but for many other infectious diseases. Up next, our 8th paper out of 10, revealing drug targets for sepsis, an association study integrating Mendelian randomization, EQTL, PQTL, and protein-protein interaction networks by Nkui Liu and Weidong Tang from the First People's Hospital of Fuyang, Hangzhou, Zhejiang, China. Let's break down its key contributions. Sepsis is a severe, often fatal condition, and finding new ways to treat it is a major challenge. This research tackles that by asking, can we use advanced genetic and protein data to uncover promising new drug targets for sepsis? To answer this, researchers employed a sophisticated approach. They integrated human genetic data, including EQTL and PQTL, with protein interaction networks. Using Mendelian randomization and advanced bioinformatics, they systematically screened for proteins associated with sepsis and explored their functional pathways. The study successfully identified 43 proteins that could be targeted by drugs for sepsis. Crucially, B3-GALT6 stood out with a highly significant association, P equals 5.24E06. Further analysis showed B3-GALT6 expression was significantly lower in sepsis patients. p less than 0.001, reinforcing its potential role. Sensitivity analyses confirmed the robustness of these findings. This research offers a significant step forward by pinpointing B3-GALT6 as a promising new therapeutic target and potential diagnostic marker for sepsis. The innovative multi-omics approach used here provides a powerful framework for future drug discovery. However, it's crucial to remember that these findings require further validation in clinical settings before they can be translated into new treatments for patients. Up next, our 9th paper out of 10, Unveiling Novel Drug Targets for Atherosclerosis, a Mendelian randomization analysis by Wenji Du and Yong Zheng from Beijing Institute of Heart, Lung, and Blood Vessel Disease, Beijing Anjin Hospital, Capital Medical University, Beijing, China. Let's break down its key contributions. Atherosclerosis remains a leading cause of heart disease, impacting millions globally. Despite advances, we still need new ways to prevent and treat this condition. This research set out to find novel drug targets by leveraging genetic insights, hoping to pave the way for more effective therapies. To achieve their goal, researchers employed a Mendelian randomization approach. They analyzed extensive genetic data from genome-wide association studies and protein quantitative trait loci. This method allowed them to use genetic variants as natural experiments to assess the causal impact of various circulating proteins on atherosclerosis risk, providing a robust framework for target identification. The study's findings were quite significant. They genetically identified causal effects of several circulating proteins on atherosclerosis risk, a crucial step in drug discovery. Specifically, lipoprotein A and proprotein convertase subtilisin/cursin type 9, or PCSK9, emerged as particularly promising drug targets. These results were consistently validated using multiple statistical approaches, strengthening their reliability. This research is a significant step forward, demonstrating how Mendelian randomization can uncover new drug targets for complex diseases like atherosclerosis. The identification of lipoprotein A and PCSK9 as promising targets is particularly exciting, building on existing knowledge and opening doors for new therapies. While these genetic insights are powerful, the next crucial step is to conduct further clinical studies to validate these targets and develop effective treatment for patients. Up next, our 10th paper out of 10. Tumor sialylation regulates G-CSF stability and promotes neutrophil-mediated immunosuppression in breast cancer by Stefan Reiter and Joseph M. Penninger from Department of Obstetrics and Gynecology, Medical University of Vienna, Vienna, Austria. Let's break down its key contributions. Breast cancer remains a significant health challenge, and while immune checkpoint inhibitors have revolutionized cancer treatment, they often fall short in many breast cancer patients. This highlights a critical need to uncover the underlying mechanisms that allow tumors to evade the immune system. Our study delves into one such mechanism, tumor sialylation. To investigate the role of tumor sialylation, we utilized various preclinical breast cancer models. Our approach involved both genetic and pharmacologic methods to inhibit sialylation, allowing us to observe its effects on the tumor microenvironment. We then employed detailed immune cell profiling and molecular analyses to uncover the specific mechanisms at play. Our findings revealed that tumor sialylation is negatively associated with T-cell infiltration, a critical indicator of immune response. Importantly, inhibiting sialylation significantly reduced immunosuppressive neutrophils while boosting beneficial CD8+ effector and memory T cells within the tumors. Mechanistically, we discovered that sialylation enhances the stability of G-CSF, a key driver of immunosuppression, and also limits the expression of MHCI, further dampening the tumor's immunogenicity. This study identifies tumor sialylation as a crucial, previously unrecognized mechanism by which breast cancer cells evade the immune system. By understanding how sialylation stabilizes G-CSF and reduces MHCI, we've uncovered a new pathway for immune suppression. Our preclinical results are exciting, suggesting that targeting sialylation could be a powerful strategy to make currently resistant breast cancers more responsive to immunotherapy, paving the way for new treatment options. That's all for today's PubMed Daily. If you found this helpful, please leave a comment, and subscribe for daily paper summaries. See you tomorrow.